leading edge of where we are in terms of understanding human memory. So I think it's a real treat that he's sharing this with us today. And Daniel, I'm really looking forward to your talk. So thanks for being here. But you couldn't have said no anyway.
Okay, so thank you both. Thank you for the opportunity to be here and presenting my work. So as Paul mentioned, I studied, I did a PhD, I just finished it in last year in Space Lab. So I have been experiencing or as a republic of BCBT for many years. So attending the conferences and listening to the speakers. So it's really an honor to be now on the other side, presenting my own work and what we have been done during the last years. So I'm going to talk about memory and how memory is represented in the mammalian brain, in the human brain specifically. I will try to present like the main knowledge we have, the main theories that we know that we have that have been important in relationship to memory, and I want to frame our results and our own research so that we can have like an overview of where we actually are, what do we know about memory in the brain. So this is the outline, so these are the three parts in which I want to share with you today. So the first one relates to the way episodic memories are formed and the idea that these are complex informations that are stored in the brain. And it is not only memory about concrete or discrete elements, but it is memories that include also information about time and about space and about experience itself. Episodic memories have been defined as these memories that enable you to re-experience the event, to re-experience it as if traveling back in time. So this is a behavioral study that we conducted last year, and I think it's interesting the results we have, so I want to share them with you. Then I will go to the intracranial in part 2 and part 3, and I want to speak about specifically how episodic retrieval takes place in the brain from a system level perspective. So I want to understand, or we tried to ask the question of how different form of representational brain statements, so the replay or reactivation of neuronal patterns that take place locally at different areas of the brain are coordinated to achieve this like complex representation of a memory, right? So what are really the timings and the frequencies in which this takes place, so that we can like look at episodic memory as more from a whole brain perspective, brain statement specific to some areas. And then I want to talk about active learning and how action and cognition affect the way we learn, we memorize, and how this is really related to hippocampal oscillations and specifically to the theta band in the hippocampus, which has been in the literature linked to many other cognitive processes. So if we start with the long-term spatial clustering. So what we know now, today, about memory is that it doesn't take place at one specific place in the brain, and that also it's not like a fixed phenomena. It's not something that memory is not stored in the brain as it can be like a document can be stored in a hard drive. You know that, right? Actually, it has some similarities to that type of information storage, but it is not the same because actually these patterns that represent memories in the brain are evolving over time, right? So they change. And this is what is called systems memory consolidation. And this has been like a key idea in the field since long, since the case of patient H10 that we discussed during this week. But starting from this case, this particular, this single case study, theories were developed, and the most important theories about episodic memories have argued that, that indeed there is this change, or this, you can think of it as a migration of signals that first encode information in the hippocampus and then are transferred to other cortical areas. Okay, so sensory information is processed in sensory areas of the brain which connect to the hippocampus and the temporal lobe. And then from there, when memories are formed, these engrams are critical to reinstate and to remember. But if this remembering takes place at a recent time point, the hippocampus is still involved in representing that information. And the reactivation of this pattern is critical. But with time, this changes because there is this process of consolidation that we know is linked to sharp wave ripples, in which the information is transferred from the hippocampus to the neocortex. And eventually, and this is recent findings, mostly conducted with rodents in orthogenetic studies, and I strongly recommend this review of the Donegagua lab. So recently it has been shown that these engrams in the hippocampus become not necessary anymore. The recalling the memory does not depend on the reactivation of these patterns of hippocampal patterns after some time has passed, because memories get consolidated and they go to the cortex. And so this uses a very strict definition of memory engrams, in which it is considered that some neuronal population is an engram of a specific memory if it can cause behavior, if it shows also perdurable changes in the morphology, so changes in spine density, number, and so on, and then that with these new techniques of optogenetics, if it can be reactivated, and it will cause, so again, gain of function, it is reactivated and it will cause the phenomena, that's a condition, and then the inactivation, it's also a condition. So if the engrams are inactivated, the experience or the memory would not be possible to retrieve in the brain, right? And so these technologies, these optogenetic technologies, also allow to trace specific neurons in the brain, right? So if you take all this into consideration and you perform some experiments, like for instance in this case using the contextual fear conditioning paradigm, you can, and you do it over time, you can exactly know which are those cells that contribute to a specific memory for fear conditioning over time. And this is how in the end you get to this diagram, because you know that if you disrupt, for instance, the hippocampal memories at recent time points, the memory is not retrieved in the rat. But if you do it at remote time points, the memory can still be retrieved. So you know that there is a migration for this memory. And this is, like, I would say, the most detailed evidence of this system's memory consolidation. And it actually answers one key question that was framed in the field of memory long ago. And it is this issue of hippocampal dependence. It shows that, at least in the rat, the hippocampus becomes dependent to retrieve a contextual memory, so memory for associations. Right? And there is some evidence also in humans, based on fMRI studies, of this strong replication of prefrontal areas, activation in fMRI with time for memory, for remote memories, and the increased activation of hippocampal fMRI, old activity during as time passes, right? But so this is telling us that the physiology of memory changes over a long period of time. But we also know, and we saw this in the presentation of Luis Pintemilla this week, on Monday, that during sleep, memories also change. Because this reactivation that I'm talking about takes place mostly during sleep. It is true that sharp wave ripples also take place in the awake state of mammals. So when, for instance, rats are just standing or deciding on what to do. But people believe that this might be for different purposes. So the purpose of sharp wave ripples during sleep is mostly related to the consolidation of relevant memories. Okay? And so if we know that during sleep there is also a change in the neural substrate of memory, and not only over months but after just some hours, we can ask if the content of these memories is also affected, right? We know that the physiology is changed, is transformed. So how does this transformation affect now the content of the memories? Okay? And this is what inspired us to perform this first experiment. And we thought that a good way of quantifying how much an associative memory or a contextual memory is transformed over time is to look at how much memory is affected by contextual, by the context in which learning takes place. So in this case, I'm talking specifically about spatial context. Right? So it has been shown previously that actually, so researchers before have in the field of memory have studied free recall dynamics in order to get an idea of what is like the structure or the organization of memory. So if you're not familiar with the field of memory, maybe you don't know what a free recall experiment is. So in free recall, this is a paradigm, like as I mentioned before, the contextual free conditioning is a very established paradigm for rats. The free recall paradigm is well established also for humans. And the idea is that you just present subjects some items, which can be of many types, and then after they studied the items, but it is true that throughout the history of the field, it has mostly been words, list of words. But then after you give some time people to study them, you don't give any instructions on how to study them, you ask them to retrieve as many as possible, as many as they can, without also not giving any instruction on how they should structure the order in which they report the items back. Okay? Then by studying precisely this order, or this dynamics, so also taking into account the timings and so on, and for things like intrusions or forgetting and so on, you can have some insight into how these memories are organized, because if you ask someone to tell you as many items he knows from a previous experience, he will be doing a process of memory search. He will search in his brain to retrieve this information, and so the order in which this is reported will tell you something about the structure. So it has been shown that many dimensions affect this structuring. Among them, time is a critical dimension, and there are classic effects like the recency effect, so the fact that you tend to recall better the items that were presented at the end of the study list, right? Or for instance, the contiguity effect, in which there is increased possibility to remember items that are contiguous in time. So not only if you remember an item, then you are more likely to remember the item that was presented just after that item when you studied it, but also the one that was presented just before the item that you studied. Okay? But time is one. There is also semantic similarity that clusters these items. And space, it's also one of these factors, okay? And this has been shown before. But what has not been shown is how this phenomenon of spatial clustering is related to is transformed over time. Right? So how much how this clustering evolves, right? During memory consolidation and for long-term storage, and also how this clustering correlates with memory performance. Okay, so is it possible, maybe, that because of so now that we have these technologies and we can like easily spatialize items or digital information in the real world, maybe this can be useful to create experiences that will optimize, learning or memory. So this is interesting to study also from a long-term perspective. And there is some evidence from neurophysiology that this happens because of a process of reinstatement or replay, neural reinstatement of the context in which learning took place. So when you are recalling the items and you're saying, yes, I saw an apple and then I saw mailbox, you are not only thinking, your brain is not only thinking about the item, but it is also remembering the place, the exact place where this learning took place. And you can see that in these curves here, where you compare how similar is the activity of a population of hippocampal neurons when you recall an item with that of the activity of the same neurons at the location where we're learning, close to the location of learning and far from it. And it is more similar, significantly more, for the location that was the same or close. And you can see that this is a very consistent effect. This is a paper that was published not that long ago in Science. What did we do? So we developed two experiments, and so it's very simple. What they have, the instructions that what they have the participants have to do. So it's first you're immersed in a virtual environment, and you have to navigate through it, and you have to find you have the instructions of finding some objects, like for instance the trash bin in this case, and then once you get close to it, you are presented with you have the instruction of finding a new one. Okay? And you do that for 13 objects. It takes more or less 10 minutes, so it's quite not long. And then you have an immediate free recall test. So you're asked to tell as many objects as you can remember. And then 24 hours we have a new test that it is a surprise test. So they don't know. They will have to be asked again about one day before, well after. Sorry. And then in experiment 2, we did a similar thing, but instead of asking for the objects, which were present all the time in the environment, we associated an image to the object that was presented only for 2 seconds, in order to avoid that this variable of presentation time of the objects, which will be different for each subject, that could be affecting the spatial clustering. And this week we increased also the number of objects, and we had 7 because some subjects had seen
Apple has.
Sorry.
And then we measure the clustering. And so the clustering is basically measured as by ranking each transition between items according to the distance. Okay, so if a transition from one item to the other is with the closest item, we put a number of 1 for that transition, for that trial. But if it is like the item goes the farthest apart, we do a 0. Okay, and then so we have for each transition a number, and then we do the mean, and so we have a mean for a session. And this is an example of how it goes in experiment 1. So you see you have like a more or less realistic town to navigate. You have to find the guitar, and you get there, and then you have the instructions of finding the trash bin. And then for experiment 2, it's the same, but when you get there, you get an image, and then you are asked to report the image and yeah. Okay, and so is it clear, like the methods and the yeah? Okay. And so we these are the results. So we found like significant clustering scores as compared to the chance level, that is at 0.05 for both time points. Okay, so this indicates that they were indeed remembering, participants were indeed remembering items according to this dimension of space and distances between objects was modulating also how they remember the items. So that means that there is, so that the influence of the context, the environmental context, is affecting this recollection dynamics. And this can be seen also in these conditional response probability curves, in which you see that you have much more probability of recalling an item that was close as compared to at a middle distance or far away. Also, we found a correlation with significant correlation with performance in the immediate and the 24 hours delayed test. So this method is interesting because it shows that the more you tend to cluster according to spatial dimension or contextual dimension, the more items you remember, not only immediately after the test, but also 24 hours later. And this reminds of this, this method of the memory palace, in which this is a mnemonic technique in which you have to associate items with rooms, and then you have to imagine yourself going through the rooms, and this is really a technique that works well, and it has been shown that this can allow people to really remember like thousands of random digits and this what is known as the memory athletes or the memory championships, in which they have to remember thousands of things, and they use this type of techniques like using space to organize memory. So that these results remember of that, but more importantly, it shows that they show that it is a long-term effect. At least it does not fade away after sleep. Right? On the contrary, it is still correlated with the performance. And we did the same. Well, these are the results for the experiment number 2, and we observed the same tendency. The results are very consistent. We also have positive correlation between the clustering scores and the number of recalled items at both time points. So another interesting result here, and this is the last one, and then I go to the next experiment, is that since we did 2 tests, we could do some overlap analysis, right? We could ask how many items were recalled at time 1, but not at time 2, or at time 2, but not at time 1. So we call them, so those remembered only at T1 were called forgotten, and then the overlapping are the ones that were recalled at the 2 moments, and then the recovered are those that don't remember at T1, but remember at T2 only. And so you see that most of the items overlap and we remember at the 2 time points. But there are some exceptions. So there are some forgotten items, and there are some recovered items. And interestingly, we found that forgotten items negatively correlated with the clustering scores, with the global clustering scores that were calculated by taking the amount of clustering at both endpoints. So this is again showing that clustering items according to space in a virtual environment that we engineered, that we designed it. So is helpful for memory 24 hours later. So this opens possibilities of to design experiences, learning experiences that could be enhance memory and learning using virtual reality technologies. Or it is also interesting, I think, to try to do this experiment over longer periods of time. Okay, we saw that memory is transformed in a period of months, and I think it will be. I would like to do this like a more time resolution, so to be able to know, to trace these curves and to see how clustering evolves over time. So I have done some pilots on that, but I don't have like clear evidence. But this experiment was done like one year ago, and then I was asking some of the subjects informally to remember again the items, and what I see is that there is. I have not conducted the full analysis yet, but it seems that the clustering is not that strong anymore, and well, of course, performance has a huge drop. But I have to still check the correlation with the performance and see if the correlation with performance is still there, because that would mean that after consolidation, so when memory is transferred from the hippocampus, which in principle should be a more associative or contextual memory, to the cortex, where you have more semantic memories, this will still. So the change in the content will also be there. But maybe the other possibility is that even when the neural basis of memory changes, you still have contextual or associative memories even after consolidation and a long time, which will be also interesting. So that's to be done. So, do you have any questions on these results? No? Okay, so we will go on then with the next part. This was a long-term spatial clustering. So now I want to talk about not memory in long term, so that long term, but more about how retrieval takes place. Yeah, and this is what I mentioned at the beginning, that we want to understand how retrieval takes place, but from a systems level. So we know that we have seen that engrams are all over the brain during retrieval. So it's not a localized phenomena, memory in the brain. So it will be interesting, and we are promoting this approach of studying the reinstatement or replay of patterns related to specific content across the brain, and in particular focusing in the hippocampus, of course, which is very important for that, but also in the neocortex, where information is ultimately stored. So, what do we know about hippocampal memories? So there is one principle that is key to understand how hippocampal memories work, and it is called rate remapping. And it basically refers to the idea that hippocampal place cells, which have, that you know about place cells, right? That have a specific firing field in the environment. Mostly, most of the cells have only one place field in hippocampus. hippocampus in CA1, in area CA1. But then these cells can, when the animal change, or when humans also have places. Let's think of the animal model. If a rat is moved from one environment to the other, it is likely to another environment. It's likely that these cells will remap. So the cells will change the position of the firing fields. Okay, and this is called like global remapping, because a completely new representation is built from different environments across the population of places. Okay, but if you do the trick of fooling the animal and not placing him from one environment to the other, like in a straightforward way, like directly, but you do some morphing between environments, between the shape of one environment and another shape, so that it's the same environment, but it just changes the shape of the borders of the enclosure. You don't observe the global remapping thing. You don't see that. You don't have like an orthogonal representation of the two environments. But you do see like a linear transition at the population level, and this is explained because cells do not change the firing field, so they still fire at the same location in the environment, but they change their firing rates. This is why it is called rate mapping. And this implies that additional information from spatial is encoded in these neurons. So when this was found, it was like really exciting for people working in the hippocampus. And then the data was explained in this blog of Cesar with John Lisman and Paul, in which they showed what are the necessary inputs from upstream regions to explain the phenomena. So they were making like predictions of what are the exact projections and the type of inputs that the hippocampus has to receive in order to explain this phenomena. And it showed that both spatial and sensory items are necessary in this proportion. So interestingly, so the point here is that you do have at the single neuron level a conjunctive code, okay? And this is a key principle of hippocampal memories. It is not only seen at the single unit level or by explaining it computationally, but you can also see the same principle when you look at human brain oscillations. Okay. So in this paradigm, here published in Life, they were looking at associative memories. So the task is to associate one item, in this case a word, with a specific color. Okay, and then there is a recognition test in which you are asked to indicate whether you were presented with the item or not. And if you think you were, you have to say also in what context, so associated to what color. Okay? So you can say that you don't know, or that you didn't see it, or that it was in red or blue. And so associative recognition would be the recognition of the item and the context. Okay? And then an item recognition could be thought as a form of familiarity, which you just know that you were presented with the item, but you don't know where exactly or associated to what context, right? And if you look at reinstatement in the hippocampus, in the human hippocampus, of oscillatory patterns, of oscillatory power, so how similar are these time frequency templates at encoding and retrieval. And you sort this by conditions, and you look at differences between conditions. You see that for associative recognition, you have higher statement. This indicates that the hippocampus is preferentially coding item context associations. Okay? This was in the literature. So, consistent with the remapping phenomena, right? But on another level of description. But now if you look at cortical representations, it's kind of different. So it's not really that much about associations. This is what was only an echo study. But you do find item-specific, so not contextual, but only item-specific range statement in the cortex. Okay, and this is, well, these two papers have like different measurements and ways of quantifying this range statement. Here it's more like a global measure, so it takes all the electrodes in the cortex, so we'll similarity measure. Here it's really more only focusing in the hippocampal electrode. And to achieve this, they increase the resolution of the representation of patterns by including more time points, more frequencies. Here they just take bands, traditional frequency bands, and the time is averaged. Okay, so these are different ways of quantifying similarity. This is cosine similarity. This is sphere correlation. These are details, but they show these two main principles, that in the hippocampus you have associative memories preferentially, and in the cortex you tend to have item-specific representations. And this is not only these two studies, which I selected now, but there are others that show that are consistent with this. Okay? So we wanted okay, no one before checked for these two types of representations simultaneously. Okay, so we wanted to see how are these two forms of local real statement in the cortex and the hippocampus coordinated, and how, from a system level, you can like out what are the timings, how can you like understand what is really happening, what are the dynamics of these forms of range statements. So we built this setup that tests basically for memory for items and memory for contexts. Okay, so we have a maze, like it has 4 arms, and then you have the instructions of going to a specific room. There are 4 rooms, and these are identified by a wall texture, so each room is different. And you have the instruction of reaching one of the rooms, and then when you get there, for each trial, we describe it like a trial, when you have to learn coding, so you just have to go to a room, and then you reach this sphere, and then represent it with one item. And then you have the instruction of going to the next room in the encoding block. Okay? You just go from room to room, and you represent this item. And then in the retrieval block, that comes like after you encoded all images, like 80 images, 20 in each one of the rooms, you have to do the same, so you still have to navigate. But now when you get into a room, you're presented with a new image. Well, you're presented again with an image that can be old or new, so it can have been presented or not. It can be like an image extracted from a new pool of images. And you're asked if you saw the image or not. So this is a spatial version of the typical recognition memory test that we saw before, but unfortunately, it's not the same phenomena as doing this. You know, notice the difference, right? This is not manipulating really context. This is asking you to associate, to report about the context, but it's not changing context. Whereas in our case, we are actually presenting these images in the retrieval test in two conditions. So one that is congruent, so the same item is presented in the same room where it was there, and the second condition that is incongruent, so the item is presented in a different room. So now we manipulate the context, and we want to see how this affects the memory for the item. Right? And so we ask also to provide responses in a scale of confidence, so that we can know. But this is also typically done in recognition tests, because like this you can, because recollection, or the process of contextual memory, typically implies that you have high confidence about remembering the item. Because if you remember the item, and you know where you learned it, you're quite confident that you did learn the item. And so this is, just for you to know how the environment looks like and how were the trials. So in this case, this texture here indicates where to what room you have to go. And so when you get into the room, you have to reach the sphere, and you're presented with an item. And then the texture changes, and you have to go to the next room. Okay. So it's and so on. So you have to do this for all units. And well, we're using video game technologies in order to make it like more valid in terms of it looks more like the real world. It's not the same as to remember an item and try to also remember the context, but it has some more ecological value. In this case, so the retrieval trial is the same thing, but now you have to respond to the question. Okay. It's clear, no? Yeah. Okay. So we did this with a healthy group of participants before going to the hospital, just to check that the paradigm works. And we found context effect in the recognition. So we have higher accuracy, so all items that are mostly recognized as old in the congruent condition, when the room and the item are the same, as compared to the incongruent or non-congruent, where we manipulate the context. This has been observed before. We also see like a tendency in the reaction times. So you take longer in responding to the non-congruent items. This is we did it in. This is not caused by any sort of bias in the responses. It's not that people are responding more often to the old condition, and you can see this in the VUCs because you see that they can perform the task with a mean that is significantly higher than chance, so they can perform the task well, but still there is this effect in the performance of the subjects. Okay. And so we validated that, we published this before, and then we went to the hospital to see how this works in the brain. Okay? So we did it with 9 epilepsy patients, but then we had to discard 2 because they were found to have epileptic seizures in the hippocampus. So we could not trust that these signals were as they are in a healthy human brain. So we discarded 2 items, and we focused specifically, we have the hypothesis that that hippocampus and lateral temporal cortex will be coordinated. Okay? act in coordination to retrieve, to reinstate these memories. So we focus for each subject on 2 electrodes on the same hemisphere, one in the hippocampus and one in the cortex. And we had patients with left and right implants. And you see that here, right? The red point indicates the hippocampus and the blue one, which are from the same patient and the same MRI scan. The blue one is in the DC of the same electrode. So, to give more details on the analysis we did. So this is what I showed you before about item context associations being reinstated preferentially in the hippocampus. But how is this calculated, like in the details? So first of all, you have to define a representational pattern. This is what we did. So we take a road trace of the hippocampal signal, LFP. Well, it's filtered, of course. Then we decompose that into frequencies from 1 to 100 hertz. We take 500 milliseconds window, and this decomposition, there are many parameters, as you know, but we chose to do it with high resolution at the lower frequencies, and then stepping into 5 hertz from 30 hertz until 100. So in total, we have a representational pattern of 44 frequencies spaced into 1 to 100 hertz and 500 milliseconds. And then, once you have this representational pattern that you can then compare, you have to slide it. We slide it over time. So these windows in steps of 100 milliseconds. So you can calculate a representational pattern that has a 500 millisecond length in steps of 100 milliseconds. And you can do that for different moments in the task. So we did that encoding and retrieval after the Q onset representation. And then these representational patterns can be compared using measures of similarity. In this case, we chose to use the Spearman correlation. We could use Pearson or measures of similarity, like the sign similarity between vectors, or the similar, depends on the assumptions you make on the properties of the data. We chose Spearman's rho because we were so the amplitude, the relative amplitude of these changes, but on the rank order. It's an unparametric measure also. And then if you compare if we have the Q onset and encoding and retrieval, and take the first representation of the pattern with the first, you would build this diagonal that you see here, right? This would be like a comparison with no lags between encoding and retrieval, right? In the diagonal. But if you take the first representational pattern at retrieval, and you compare that with one of the last representational patterns at encoding, you will end up off diagonal, right? So this will be a measure of lagged correlation. You are comparing early retrieval time with late encoding time. So we add this dimension of time in order to compare all possibilities in the encoding and retrieval epochs. Okay, so is that clear, the restatement analysis? Yeah. So we'll go to the results now. So this is the first thing we found, which is consistent with what I showed you before. So we have a reinstatement of item context associations in the hippocampus. And this is seen in the fact that we found increased encoding retrieval similarity for items encoded in the same context as compared to different contexts. This is the reinstatement map that is built the way I showed you before, for congruent items, so those items encoded and retrieved in the same group. And this is the one for the incongruent one. So you see that you have this stripe here locked to the onset of the stimulus at retrieval. You have like a stripe of increased similarities. If you do the comparisons between the two types of trials, you see that this is statistically significant at p at 0.05 in this cluster here that starts at around 1.8 seconds in encoding time, but just locked to the onset of retrieval. So you are actually reinstating more strongly activity of late encoding, early at retrieval. This is what this data is showing at least. Although we do see that there is maybe it doesn't reach significance, but we do have some clusters here. We observe that if you compare room now, irrespective of items, so all items encoding in one room versus all items encoding in a different room, versus all items that were encoded in a different room. Yeah, you see that if you just take into account the room information, the hippocampus is going to be active also. So it appears that this dynamic pattern of oscillatory power that was calculated, as I explained before, is only happening when there is a match between items and context at encoding and retrieval. And if we look at the specific characteristics of the cluster, the significant cluster, we can see that it is advanced in time because we are reinstating activity of late encoding, early at retrieval, and it is also time warped, elongated in time in the sense that it is not on the diagram. It's not. It doesn't have a shape that is elongated on the diagonal. So it has some sort of lag. So if we focus specifically on this cluster and we quantify how much reinstatement or encoding retrieval similarity happens in congruent and incongruent conditions, we see that it is quite consistent across subjects. Each line is a subject here, and that it is different from zero, from chance, in this specific cluster. But it is not different from chance in the incongruent condition. This is the specific values for this cluster. And it tells us that the effect that we first saw is related to the increases in similarity for congruent and not significantly high decreases in similarity for incongruent. Okay, but as I mentioned that we wanted to study the coordination of different forms of local reinstatement. So we have to look at the cortex also. And we can do the same analysis here, but focusing in the cortical electrodes. And so we found like a completely different pattern of responses, which is interesting because we were actually measuring from the same trials. So these are the same trials, same subjects, same times. So these were simultaneous recordings. And the patterns are completely different. So what we observe in the LTC, the lateral temporal cortex, is reinstatement of item information. And we observe that in two clusters that according to our criteria, were considered significant. And if we look at the details, as we did before, we see that encoding field similarity is higher than zero in the same item, but not in the different item in cluster 1. And the same pattern is happening in cluster 2. So it is interesting to see that the timings are shifted. Right? So we have hippocampus like just locked to the onset at retrieval, and the cortex coming after that, and retreating after the time. Right? So if we do the congruent versus incongruent, so the same as the one which we saw differences in the hippocampus, we don't see anything here. So the cortex is not really caring about associations between items that are in context, or only about the item information. Right? Same if you do the same rule versus different from contrast. So it's not only about context, but more about the items. Right? And one, so we have this double dissociation between hippocampal and cortical reinstatement in terms of space, so hippocampus and cortex, and time. Right? But importantly, what we also found is that this reinstatement is functionally relevant because it correlates with these judgments of confidence that I explained before. So in this contrast in which we compare high confidence trials versus low confidence trials, we do observe a significant cluster which shows that you have increased similarity in the ATC for high confidence trials, significantly higher than 0 compared to low. And this cluster that you see here comes right up before participants respond. This is the mean response time. Standard error. So the functionally relevant cluster in the LTC, it happens before the participants respond. So because memory test, right? And what is also interesting is that it partially overlaps with the item-specific cluster we found before. So it's around the same area. But this contrast is built with different trials, right? But you have the same, overlapping cluster. So this suggests that maybe it is the quality of item-specific reinstatement that modulates confidence, right? So when we calculated, I can do the results here, but we calculated high and low confidence reinstatement in this cluster, first identified the item contrast. And when we compare those conditions here, we found a very similar pattern of result than this. So this is comparing in the confidence cluster, but when you compare high and low confidence in the item cluster, you do find the same pattern of result. So this indicates that it is actually this reliability of this information here that modulates confidence. And we didn't find anything related to confidence in the hippocampus. But so we have, until now, what I've shown is simply that we have reinstatement of item complex associations in the hippocampus and reinstatement of items in the cortex, that they have different timings, and that the LTC item reinstatement seems to be functionally relevant. Okay? But if we go into the details, we can also ask the question of what are the contributing frequencies to these patterns, right? And there is some theoretical reason of doing that, and it is related to the fact that low frequency oscillations are known to have many cognitive roles in cognition in the mammalian brain, and the role of low frequency oscillations in the cortex is not as well characterized as in the hippocampus. And when we build these representational patterns that I was showing before, just by including information in specific bands, like this traditional band, but this is like the delta theta, right? 1 to 8 hertz. we found the same effect in the congruent-incongruent contrast in the hippocampus, here. Right? But when we build it, by taking into account only alpha, beta, or gamma, you don't see reinstatement happen. So, hippocampal reinstatement seems to be dependent on low frequency oscillations. Okay? In the cortex, on the other hand, for the same item versus different item, or for the high confidence versus low confidence contrast, you need all frequencies to get the pattern of results that we previously saw. So this relies on broadband frequencies, but the hippocampal reinstatement is relying on low frequencies mostly. So now we have a triple dissociation, not only in space and time, but also frequencies. Right? So these are well distinct physiological phenomena, right? But they are looking at the same trials and the same items simultaneously. We are characterizing different forms of reinstatement. And when you do a jackknife, this is to validate these frequency findings. When you do a jackknife procedure in which you just take out now the 1 to 8 hertz band and calculating reinstatement, and you subtract a reinstatement calculated without the 1 to 8 hertz frequency to that to reinstatement calculated with all frequencies, you see that the reduction is much higher in the hippocampal congruent-incongruent cluster as compared to the clusters, all clusters in the LTC, which seem not to be modulated by the disruption, but by taking out this band. Okay, so I think these are nice and consistent results with two different methods to show the same thing. But then we come now to the critical point, because up until now, okay, we characterized a little bit more how reinstatement happens in these two areas, but this was, in a sense, suggested by previous literature. Okay? So the critical thing was to see how these two forms of local reinstatement are coordinated and interact from a systems level. Well, the fact that we saw different timings also tell us something about that. But we wanted to see first if the level of reinstatement in the two areas, with different timings and different representational contents, was correlated across trials, right? So for this, the first thing we did is the simplest. We just correlated mean reinstatement in the hippocampal cluster and mean reinstatement in the LTC cluster across trials. And this gives us this plot here, in which, well, when you take into consideration all trials, not only those congruent or incongruent, you see that you have a significant correlation. So this indicates that those trials in which you have high levels of reinstatement in the hippocampus are the same trials in which you also have high reinstatement in the LTC. Okay? So this suggests like a synchronized or coordinated phenomenon. But now, is this phenomena specific, or is it happening like in general at any of the encoding and retrieval time points? Right? Or is it specifically to the moments in which you have high reinstatement of the particular contents that are represented in the hippocampus and the LTC? This is what we test with this analysis, which is an analysis that we developed, we implemented, and has not been used before. But basically, we were using one of the two clusters as a seed in order to compare at the group level correlations with all the encoding retrieval time space of the other region. So we took first the hippocampal cluster as a seed, and we correlated with all the encoding retrieval time time space using a similar window as the other cluster, the other regions cluster. And we do observe that there is some overlap in the increased correlations. So those regions in which you have increased correlations overlap with the cluster that was where we first observed this correlation. So increased misstatement in the LTC. And if you take the other LTC cluster as a C and you correlate with all the encoding retrieval time space in the hippocampus, you observe also that there is an overlap, and that it is not only, well, in this case, it's a little bit more broad, so it covers also moments in time that are happening before encoding time, as compared to the cluster that we identified before. But this cluster here overlaps with this one part here. But anyway, what this is showing is that it is not a general phenomena happening all over the time space, but it is more close to the moments in time in which we have increases in this similarity of the specific representational contents of each region. So then, after this, we wanted to see how this relates to. So what are the mechanisms, right? So we observe a correlation. In a sense, it's just a correlation, but we want to know if this is related to known mechanisms of information transfer, right? Because if these things are happening in coordination, it must be because in some sense they are communicating. So we looked at phase synchronization, because phase synchronization has been theorized to be important for information transfer. So a mechanism that allows for information transfer in the brain. And we now correlated levels of phase synchronization with levels of hippocampal brain statement. We did it for the hippocampus and for the LTC, but here I'm showing the LTC because this is where we found something. So we did it for all frequencies, like you have all frequencies from 1 to 100 here in the x axis, and we took the mean of well, not the mean, it's not the mean, it's just the phase synchronization in this period from 0 to 500 milliseconds, and we correlated that across trials with the levels of the hippocampal brain statement. And we observed that there is a significant cluster, which was the highest in the when we did the correction for multiple comparisons, so it's strongly significant. From 73 to 80 something hertz. I think it's from 70 to 83 hertz. So gamma synchronization correlates with levels of hippocampal brain statement. But this gamma synchronization takes place just after the onset of the stimulus at retrieval. So you're presented with the stimulus in the test phase of the experiment, you see it, you have some gamma synchronization, and then after that you have some increased in brain statement, and after that you feel confident and you say, yes, I saw it. This will be more or less the time how this event takes place. And then we did it for all encoding retrieval time space until the offset of the LTC cluster here, because this is where information transfer should happen. Like, just after the statement, the representation of this information is high. So before that, so we convert neurons, and we observed again a significant cluster that also survived for multiple conversions here, suggesting the same, so that these two mechanisms and phenomena are also coordinated, so that you can predict how much reinstatement you will have in the LTC if you only look at the synchronization between these two regions in an earlier time window. Yeah, so to sum up, so what we found is a triple dissociation of hippocampal and LTC reinstatement in terms of representation of content. So item context associations in the hippocampus and item-specific information in the LTC in terms of relative timings. So hippocampal reinstatement taking place before, just log to the onset at retrieval and until 0.5, 0.6 seconds, and the LTC happening after 1 second. And in terms of the contributing frequencies, so hippocampal reinstatement relying on the low frequencies 1 to 8, while LTC brought back. We also found a functionally relevant, that LTC reinstatement is functionally relevant, and that correlates or depends on the reported confidence. We also found that these two forms of reinstatement, if you look at them from a system level, are coordinated across trials, not in general, but specifically in those moments in times in which they are high, and that LTC, so neocortical reinstatement, is also correlated with early phase synchronization between the two regions. So this shows that, yeah, it's consistent with what I showed at the beginning, that memory is not only built it's not localized in the brain, but it takes place in the whole brain. And we are measuring here in the LTC, but of course, we expect to see that any region that is important for representing the specific items that are memorized at the beginning will show similar patterns of interaction with the hippocampus. Is that right? This is visual stimuli, so the LTC is involved in the representation of visual stimuli, but if it was from another sensory modality, we would expect to find these interactions also similar to this. So this is, I think we covered the second part of the talk. So this paper is under review now, so let's see what happens with this. Now I want to discuss some more preliminary results that we have also in another interpreted setup. So we can finish. Well, we're doing in time.
How long do you need?
I don't know, not much.
No, that's fine.
Say how much, because we're
I can do it 15 minutes maybe.
Okay, go ahead.
If not,
just
Go ahead.
Just with the idea.
Okay. It's going to be short. So here, the idea is to see how, what are the neural correlates, or how we can explain the benefits that we have observed in active learning versus passive from a neurophysiological perspective. So we know that active learning helps memory. So this was shown, for instance, in this paper, in which people study image arrays in active and passive conditions. So the task is to explore this 2D space, so it's a screen, which is overlaid by a blur, so you don't see what's behind. But you can control well, in one of the conditions, you have this window that lets you see through the group. Okay? So in one of the conditions, you can control the computer mouse in order to move the window, and you can see what is behind. And in a second condition, you are observing the trajectory of another subject. Okay. And the instructions are, okay, explore and learn as many items as you can. You see that the results, behavioral results, show that you tend to recognize in a recognition memory test afterwards much better items that were studied in the active condition versus the passive one in which you're just looking at the trajectory of the interactions of another subject. So this was the inspiration for us to build this setup in which we were doing the same, but now in a three-dimensional space. So you have these red boxes on the ground, and you can navigate here. We are actually developing now an embodied version of the task, which I think it would be interesting. But this is a desktop version because we wanted to run it in the hospital. So you can navigate this square virtual environment, and you have the instructions of these being called boxes and number of items, and you do that in an active, volitional, and also in a passive condition in which you just look at you observe the trajectory of a previous subject. You have blocks of exploration of 3 minutes, and then a pause of 20 seconds, and then 3 blocks active and 3 passive, which are randomized. And then this shows that, well, this has nothing to do with previous, with most memory studies, because typically in a memory study setup, we just present the item for a specific time that is controlled, and it's the same for all subjects. But here, you see this is a trajectory of one subject as an example. You can decide to revisit items, and you can study them as long as you want, for as long as when you decide the timings and the items that you will observe, right? So on a moment by moment basis. So this is like a different way of studying memory. And then the question is the same. Have you seen this typical recognition? So have you seen it before? And there are some new images also. Yeah. So we did. We did with healthy subjects and with patients, with 11 patients in the hospital. This is a brain cube representation of the data set. So this is a timeline where we have so the circles represent when the person entered into one of the boxes, and the lines are when they go out. We have this we have explored this in the SIM also. And this is a video of the task. And this is all the electrodes from one subject that we are correlating in time in 500 milliseconds beams that are sliding in 100 milliseconds. And it just shows the complexity of what's happening in the brain of this person while performing the task. But we haven't linked this to behavior in any way yet, though we are running some criteria analysis to see if we can extract something meaningful from this high-dimensional data. But the idea is that you see that we have the traces over time, we have the markers. So if you look at here, this is showing when you enter and when you go out one of the boxes. We know what the person is looking at every moment. We know if the item was recalled or not, and we have all this complex data that now we have to make sense of it. But we had, when we run the experiment, the hypothesis was that we will find differences in the exploration for active and passive. But we first ran the protocol with as in the followed same methodology as in the other experiments. So we did it before with healthy human subjects with 23 healthy human subjects, and we did find the same effect of volition in the old endorsement rate. So items that were correctly recognized as old in both conditions, because in both conditions items were old, but the rate is higher for volitional, and we also found increased confidence for volitional. So it's like you feel more confident when you actually control the navigation. We saw that this effect is also not related to the, well, it's not only due to noise caused by changes in exposure time to items, because for attention, because we see that for items that the effect is even higher when you look at items that were exposed for long times, which is consistent with this previous study I showed before, that the effect is increased for items that are explored long. And we excluded those items that were explored for very short times. But if we look at the, so this study also did some fMRI and the same study with the same protocol with patients with amnesia. And they found that the effect is not there when you have hippocampal damage, so you don't have an effect of volition when you don't have a functioning hippocampus. And they also found, using fMRI, the limitations in time resolution that has, that there is like a coordination of the hippocampus with some critical regions for memory that are increased for volitional versus passive. So we had this idea in mind, and we wanted to observe how this now, with the resolution of intracranial EEG, we could observe differences in the hippocampal electrodes, and so this is just a hint on that, and it shows that if you take activity from all navigation blocks and you compare those active versus those passive, you see that most of the frequencies from 1 to 100 Hertz are not affected by this difference in navigation, but the low frequencies, so from 2 to 7, cluster into a so a significant cluster of differences. So this is telling us that so that theta in the hippocampus is playing a critical role. So we now not only know that hippocampus is more involved in this form of active learning because of fMRI that we knew before, but we also know the specific frequencies in which that effect is observed. So it's in the lower frequencies. It's a nice result, but of course we still have to unpack this and take a look at the details. And we are doing that, although this is some preliminary results. But here I'm using like a global measure of neural similarity, and I am using a jackknife procedure just to select what are the most informative electrodes, and I'm comparing recollection versus familiarity, so those items that are recollected with high confidence versus less confidence. And if you select the most informative electrodes and you run the analysis again, you see that this memory engram, we call this memory engram, is detectable. It's very clear, and you can very reliably predict or know what type of trial this was, if it was a recollection trial or a trial in which recollection did not take place. And this jackknife procedure will also help us to look for item specificity. But this is not taking into account all electrodes and not only the hippocampal one. So I think this will be interesting to follow up and also to approach it from a machine learning or computational point of view, because probably we can using or doing this network analysis in which we can get more insight into the global mechanisms or what is happening across all the electrodes and how the information is represented here. So yeah. So this is it. So thank you.
All right, so now you know how the human brain stores and recalls memories, and it's a pretty involved kind of form of analysis, as you might have noticed, right? And it takes a while to get your head around. So I would be really surprised if you got everything in one shot. So in that sense, young people must have a lot of questions. And so what Daniel really shows is that the information that you store in the memory of the hippocampus and the lateral or the temporal lobe, so it's sort of expressed in patterns of frequencies, right? So you have, if you want, you can imagine different oscillators. They oscillate at different frequencies, and then you make different combinations of these oscillatory patterns that sort of represent the content of memory. That doesn't mean at a single cell level that's how it's done, but this is at the sort of, if you look at how volumes of cells respond, this is now your echo of information as it is stored in the hippocampus, in the cortex. But then what Dan also showed is that the hippocampus is not really resonating, if you want, with the items, the actual elements that you store, but it's more resonating with their relationship, right? And the actual item information that sits more in your hippocampus, which goes back to a model by people like Eichenbaum, right? Standard models of memory that really see this sort of a networked phenomenon, where a hippocampus is more like the orchestrator and orchestrating the actual information that hangs together. Like in this room, there are many elements in this room, and the state representation of these elements sits then more in the cortex, in the temporal lobe, while the hippocampus has set the pointers to these elements and brings them together and say, these guys belong together, and now you have the room, right? So that's one of the key observations, and then what Daniel also shows, and that's of course a logical question, but then how does this whole thing hang together? Right? How is the information then exchanged that these pointers can actually do their job? Right? So there are two things that are important there. That hippocampus has sort of the orchestrator of the memory is time warped in a way that it moves forward in time when you have to recall. Right? So it has a very quick response, as you saw in this first experiment in human subjects, that already as soon as the cue comes on that you have to recall the memory, you see the hippocampus kicks in much earlier than it did when you were storing the pattern. Okay? The cortical response is shifting in the opposite direction. It comes later in time. Right? So there's time warping in both directions in these two structures. But that first hippocampus comes on and it starts to talk to the cortex to sort of coordinate its memory. And then you see that this happens in very distinct frequencies. Right? So there are, if you want, certain communication channels defined in this frequency space that play very distinct roles in how the contact is recalled, but also how the coordination is then set up between these structures. So this is some of the fundamental observations that Daniel now gave to us, which means the language of the brain, and as far as we can infer that from the measurements we make, and with these local field potentials, is sort of sets of oscillators that are being bound together in their phase relationships. So this is what it looks like. So this is sort of a bit like a summary to also sort of give you a bit more time to think about your questions. All right, question time. Joshua.
So you basically start using metaphors from physics. You basically have the theta field, the gamma field, and so on. In this field you have different wave patterns, and if they're stable, they're probably something like solitons, and the individual neurons try to entrain themselves with these patterns in order to become part of a representation. Now, a simple question might be, how does working memory work? When I was young and innocent, I thought it's more like a run, you need to constantly refresh it, and if you basically go to sleep for a while or your activity lapses, you lose the representations because they're not constantly refreshed and your wave patterns die down. Now, observed in several patients during surgery that they seem to have not lost the memory state that they were in before they went into anesthesia. For instance, my son had surgery and he was choking and gagging on the anesthetic gas. And 6 hours later, he woke up in a different context, but started choking and gagging and told us that the anesthetic didn't work. Basically, there was no loss of the in-between working memory state. And I wonder how it's possible that working memory is conserved for such a long time if this is done in wave patterns and oscillators, right? There must be some state that are beyond the oscillation. How would that work?
Yeah. Well, the mechanisms in which of how working memory is transformed to long-term memory are not really that clear, right? But we know that working memory is basically and very much dependent on theta gamma coupling, well, that is also a way of working for long-term memory. How this transformation of this happens from working memory to long-term memory happens is not really known. On the other hand, well, what we do know is that memories that are stronger get consolidated by this mechanism of sharp wave ripple and this replay. So this phenomena or this case you're mentioning, I wouldn't have an idea of how to explain it, like, because it's like a complete switch off and switch on, like a reset of the state, like if the state would have been set in some way in the memory code to then retrieve.
I don't think it was long-term memory. I think it was still working memory. I suspect that the working memory state was preserved during anesthesia and not transformed into long-term memory. It was just basically frozen in time and then continued, put on pause.
Yeah, but then you can also speculate a little bit more, right? Because your results make, I think, a very strong suggestion how the memory is stored in a structural form, right? What its properties should be.
Well, it should be like if that is a real case, it should be like an index, right? Linking the hippocampus with the cortex, right? So this.
No, but not only that. Also, you can already say from your data, also to answer Joshua, that the item information, at least, is stored in small oscillatory circuits that have different kinds of resonance frequencies.
Yeah.
Right. And that they're linked together through hippocampal pointers. So from that you would suggest I can use small cortical circuits in cortical columns that dependent on transduction delays, for instance, between the excitatory elements, will have different resonance frequencies. This is how they will shift through the different gamma range, right? This is how you can configure that. Some of these guys get strengthened. This is now my item information. Right? And hippocampus will set up links, which probably is a synaptic connection, right? To that circuit. And then within hippocampus, you would use the rapid plasticity in CA3 to build an attractor that puts these pointers together. What's wrong with this speculation?
No, that is, well, the traditional way of thinking of long term. Right? But this is.
No, but still, to stable. I don't care if it's long or short term. You must stabilize it structurally because the thing is switched off, right? This brain was switched off.
Yeah.
So there must be the structural
The long-term memory of working memory.
Yeah, whatever, yeah. Look, these categories are very coarse, and it will be much more gradual in the brain anyway,
Sure.
right? So but you can switch off the brain, and you turn it on, you turn on the thalamocortical system again, and up there come the oscillators, and they're still in the same state.
Yeah.
You must have a structural.
So you're suggesting it's done in the hippocampus. The hippocampus basically holds the scene graph as objects, and the neocortex implements the representation of the objects in the
That's right. That will be the idea.
Yeah.
Other questions? Yes, please.
So maybe I missed that, but one question for the second part. You was talking about clusters, no?
Yeah.
if you can go back to the time matrix of the 751, yes, that one. Okay.
This one?
How the cluster you were talking about are defined? So I was thinking about how you define the cluster, no? Maybe this cluster can be more wide or narrow or...
So this cluster is defined simply by taking all encoding retrieval beans that you have in this map. So that's value. Okay. And taking also all of this second one set of trails and doing a t-test for each one of them, 4 of them. Okay, so that will give you a significance value for each one of the time beans, the encoding retrieval.
Okay,
time pairs.
There's no hierarchical clustering or clustering algorithm that you performed on the matrix.
No, I have the same problem. He used this word cluster, but it means something else. Then it just means...
No,
for you it means something... It's not cluster analysis, right?
No, but this is called cluster. It's not that I invented the term cluster for this type of analysis.
Yeah, but if you walk into the room from a background and they say other kind of statistical analysis...
This is not a genetic clustering algorithm or whatsoever. It's just a t-test between all time pairs, and then you group them just based on the contiguity of these time beans. And then you build a...
It's a group of correlations that are significant, essentially.
No, it's not a correlation, it's a group of p-values that are significant.
Okay.
Right, okay. P-values, yes.
Can you do one question for the third part? No? The correlation matrix, one of the last correlation matrix that you showed us, the U1, I don't know if you can go on the last experiment.
Which one?
And the third part of the talk, one of the 3D.
Yeah.
This one. Okay, this is from fMRI data or LFP? I don't
no, it's built from these saving traces here.
Okay, that are LFP.
LFPs,
okay. Okay, and so why just positive correlations and not negative correlations?
Yeah, no, I mean, you could build these things in many ways. I just put it here to show how complex the data is, in time and so on. But of course, here again, some decisions that are just arbitrary. They could also take negative
not
ones.
thresholding on the matrix or something like this.
So yeah, I did threshold it here, but not for a specific reason.
Okay, because you were talking about graph analysis, and so
Yeah.
I was thinking what kind of network analysis you were thinking about, and there's a connection with just taking positive correlation and not with the spectrum.
Actually, for the graph network analysis we have been working on, we have built positive correlation matrices, and we are looking at differences between them.
So separately,
to build the graphs
positive and negative networks.
Yeah, we are doing that, but it's not telling us much for the moment, but we are following that strategy.
For curiosity, what kind of network analysis are you performing?
So we are looking at basic things for the moment, but looking at where are the hubs, what are the connections, more strong connections, what are the centrality of the network. So also in collaboration with Cerces is doing some complexity analysis based on this measure phi over time. He has developed in the lab. So this is more or less what it's still preliminary.
Other questions? All right. If not, thank you very much, Daniel.